Right. Obviously, various options are provided as possible answers to the following questions. Choose the answer and write only the letter A to D next to the question numbers 1.1.1 to 1.1.9. Write in the answer book, e.g. 1.10D. Okay. 1.1.1 is about Lamarckism, or what you call the Lamarck's theory of evolution, which basically has three main parts. I'm going to first mention that what we call Lamarckism first, so that you can just be able to relate to this. Right, Lamarckism. Okay, in short, Lamarckism has three main parts. It indicates that the living species were different from those found in fossil records. So it compares living species different from those from fossil records. So one looks at now and the past, one realizes that according to Lamarckism or Lamarck's theory of evolution, the living species are different from the fossil records. Number two, domestication of wild plants and animals and selective breeding resulted in plants and animals changing. In other words, domestication obviously has to do with making these things domestic and bring them home from the wild. So domestication made plants and animals to change. Okay, so point number three, according to Lamarck's theory of evolution, crossbreeding, plants often led to new characteristics appearing in the offspring. So in other words, crossbreeding is the third point. Right, so crossbreeding plants often led to characteristics appearing in the offspring. Okay, new characteristics. So offspring meant or implied new characteristics. Right, new characteristics or just some new properties in the offspring. Characteristics in the offspring. Okay, so this is what we call Lamarckism, or what we call in the offspring or offsprings. Right, so now at this point, we need to choose according to these 3 points of Lamarckism or Lamarck's theory of evolution. And if you look very carefully, we're able to see clearly what is happening here. Right, so we need to study this very carefully and answer the questions that follow in 1.1.1. And now it is very clear at this point that organisms evolve because they adapt to their environment. And according to Lamarck's theory of evolution, so that becomes the answer to the question. So it is actually option B, for example, because you can see that the living species are different from fossil records. So Lamarckism actually says that there's always a change. These things change. The crossbreeding plants will actually develop new characteristics, and the domestication will actually make the plants and the animals to change. If you take them from the bush and you bring them home, then they're gonna change. The dogs that were in the bush and now they brought home, they actually therefore undergo some change. Now, the notion of the genotype for a specific characteristic in 1.1.2 is also very, very significant, right? The genotype for a specific characteristic has what properties? Does it contain 2 chromosomes? Is it the physical appearance of an individual? Is the composition of a gene pair? Is represented by one allele? So obviously in 1.1.2, you can see that now the genotype for a specific characteristic, right, is actually is the composition of a gene pair. It's the composition of a gene pair. That is what we call by the genotype of a specific characteristic. So it's the composition of a gene pair. Right. So yeah, I'm just going to mention this because it's multiple choice, so you just take note of these things, and then they can help you when you are in the exam. All right. In 1.1.3, it's important to note a couple of things like after the discovery of a fossil, scientists classified it as an African ape because it had what? After the discovery of a fossil, there was the classification of the fossil as an African ape. Why was the fossil classified as an African ape? Okay, so obviously we're looking at 1.1.3 now, and the correct option is, I'm not going to really waste a lot of your time. The reason was because of it had a small cranium, and a prognathous jaw and large canines. So that is the correct option there. That is why it was called the African ape. So I'm just going to mention this because you guys are writing in a few minutes. So the African ape is because it had a small cranium and it had actually a prognathous jaw and large canines. That is why the fossil was called an African ape. Okay, we're just discussing some of these so that you can remember them in the exam. The idea of what to call the statements below describe the steps in the process of cloning an animal. Okay, there's something called cloning an animal. So note that one. The embryo is implanted into the uterus of an adult female for development. The nucleus from a somatic cell of the donor is extracted. The nucleus from the somatic cell is inserted into the ovum. The nucleus from the ovum of another individual is removed. The ovum with the new nucleus is given an electric shock to stimulate cell division and formation of the embryo. All right, so which combination shows the correct order in the steps? Okay, obviously we're just discussing briefly so that you can have an idea and get one thing or two here. So obviously we'll understand that in 1.1.4, what is very important is that now this is which combination shows the correct order of the steps. Which one shows the correct order of the steps? So in 1.1.4 would understand that first things first, you'd have that the nucleus from a somatic cell of the donor is extracted, number one, okay? So in other words, you can see that the correct order will sort of be that this one is 1, right? And then after that, you would look at the fact that number 2 would be what? The nucleus from the ovum of another individual is removed, 2, right? So 3 would be actually the fact that the nucleus from a somatic cell is inserted into the ovum. That would be 3. And then 4, the ovum with the new nucleus is given an electric shock to stimulate cell division and formation of an embryo. And then we have 5. Right, the embryo is implanted into the uterus of an adult female for development. Okay, these are the process. These are the steps that actually involve the process of cloning an animal. How to clone an animal? We start as follows. We notice that the nucleus from a somatic cell of the donor is extracted. So you extract the nucleus from the somatic cell of the donor. Number 2, the nucleus that we extracted. So first we extract the nucleus. The nucleus that we extracted from the ovum of another individual is obviously removed, right? And then now you note that number 3, the nucleus from the somatic cell is inserted into the ovum. And then now number 4 is the ovum with the new nucleus is given electric shock to stimulate cell division and formation of an embryo. That is 4. And then 5, the embryo is implanted into the uterus of another female for development. So in 1.1.4, right, the correct option will have to be option A. All right, so just note that. Right, so we're looking at this one. the notion of what you call the shrubs, right? So this is about the shrubs of the family Proteaceae of the waratahs and proteas can be found in Australia, South America, Indochina, and parts of Africa as shown on the map below. Okay, so obviously we have South America, Africa, and Australia, as mentioned here. All right, South America, Africa, and Australia shown. Okay, so yeah, it is hypothesized that all continents were once large continents called Pangaea. So you had obviously Pangaea as the fact that the continent was in the beginning, you had a large continent, and that they separated due to continental drift. So there was continental drift, and then the formation of small continents. Now, like you have Africa as a continent, and other continents which involve Asia, and like they say, South America, Africa, and Australia, they now like separate, and you can see the ocean between the continents. Okay, now what is the evidence of this? The evidence that the family Proteaceae, right, so what is that evidence? So obviously I'm not going to waste a lot of the time on 1.5, but the evidence is that the evidence arose from a common ancestor when Pangaea is separated. So this one is theory, so you need to know it by actually the fact that how the Pangaea story. What is the Pangaea story? The Pangaea story is that the world was one continent. It did not have the ocean. It did not have, in other words, Africa and South America were one thing. They were like joined together. And Australia and Africa were joined together. There was no ocean between them. But now the ocean exists. Why did people say that? Because many people, when they were born, they saw South America and Africa separated like this. And they saw Australia separated by the ocean. The evidence arose that the family Proteaceae arose from a common ancestor when Pangaea separated. Okay, so yeah, that's the thing. So common ancestor is the answer to this. Okay, right, let's look at the girl and the mother and the males, because now the diagram below shows the DNA profiles. So looking at the DNA profiles, very interesting. The deoxyribonucleic acid of a girl, her mother, and 4 males. Here's the girl and their DNA profile, that of the mother. The 1, 2, 3, 4 males, each one has their DNA profile. You can see the DNA profile of each of the males is different. Now, if you look at the rest of this, which male is the girl's biological father? Let's look at that. Which one? So you just have to compare in 1.1.6, which one, because now which male is the girl's biological father? The mother is this one, rather the girl is this one, the mother is that, and the males are this. We don't know who the father of the girls. In other words, the male who is the girl's biological father. Who is the father here amongst the 4? So you think about this in 1.9.6. Right, you'll see that in this is the girl. And now you need to compare the males and look at the profile that is similar to that of the girl here. Okay, and if you look very carefully at 1.1.6, right, 1.1.6, you realize that you have that option number, letter C, number 3, profile 3, the male 3 here. Okay, is actually the girl's biological father. Why? Because you really can see that there are most certainly greater similarities there. Okay, so note that exactly. So if you look at the girl, the mother, and then now you actually put together the DNA profile of the girl and the mother, and you want to compare with that of the males, because 1, 2, 3, 4 different males, you're able to see that it's actually male number 3 that combines both the DNA profiles of the mother and the girl, to yield. So in other words, we can link this male person to be the biological father of the girl. Okay, so note that DNA profiling there. Okay. The following characteristics of a group of animals. Okay, let's get the characteristics. Able to interbreed, occupy same habitat, produce infertile offspring, okay, belong to the same species. Now, which combination correctly represents a population? Okay, now this is like population dynamics. Population dynamics is actually highlighted by 1.7. So which one will be actually correctly representing a population? So in 1.7, you actually can see that now it is first, if you look at this here, it is the following, right? You are able to interbreed in a population. We understand that obviously the species are able to, or the group of animals are able to interbreed amongst each other. So, but also they occupy the same habitat. The population, like people and the animals, they occupy the same habitat. Like, well, they live in Soweto. The people live in Soweto. Some people live in Soweto. So you can really see Soweto as having a population. Okay. Moreover, you can see that they belong to the same species. They belong to the same species. The group of animals is a group of animals, not the group. Normally, they belong to the same species. So, in other words, the correct option in 1.1.7 would have to be option B. Okay. Obviously, we agree that it's a group of animals, or you can even look at a group of people. They live together. They're able to interbreed. They occupy the same habitat, the same space, but also they belong to the same species. But producing infertile offspring is not really the characteristic of what? Of a population there. So Roman figure 3 is out. The infertile offspring, it's not one of the options. An infertile offspring would just be that offspring that is infertile in the sense of not really able to reproduce there. Okay, if you look at 1.1.8, during anaphase 2 of meiosis, the two chromatids of a chromosome are pulled apart. Okay, this is interesting because they are discussing what is happening in anaphase 2 of meiosis. The two chromatids of a chromosome are pulled apart. Pulled apart, two chromosomes, the two chromatids of a chromosome are pulled apart, each moving towards opposite poles at a rate of 1 micrometer per second. The distance in micrometers between the chromatids after 20 seconds is what? Okay, let's look at this question. Obviously, in 1.1.8, you need to look at that and think. Okay, but yeah, you need to look at the distance. So distance in micrometers between the two chromatids after 20 seconds is what? So obviously at a rate of these per second. Okay, so obviously it's going to be option D, which is 40. How do you do that one? Right, you need to look at the fact that you need to calculate the distance. So you need to use this formula from grade 10 and say speed equals distance over time. And so if you want to get speed is distance over time. Okay, you can use physics. You do physics here. Okay, so you can do time, for example. Time is distance over speed. You can do distance. Distance is speed times time. So distance equals speed times time. So obviously, because there are two of them, you can use just a physics notion. Because what is the speed? Opposite poles at the rate of 1 micrometer per second. So you need to look at this now. Okay, it's also in micrometers, this, and the micrometer, okay, it's physics, like the micrometer would be like this, which is times 10 to the minus 6 meters. Okay, need to mention we just need the distance. So the distance is speed times time. So you're gonna find the distance between the chromatids because each one is 1 micrometer per second. So you're gonna find like distance 1 plus distance 2. So right, distance 1, distance 2, which is speed times time, plus speed times time, like speed 2 times 2, speed 1 times 1. Okay, now you just do the calculation here. What is speed 1? Okay, but obviously we understand that the two chromatids of a chroma are pulled apart, each moving towards the opposite poles at a rate of 1 micrometer per second. So the speed is 1 micrometer per second, and the time is 20 seconds. But also this one is 1 micrometer, 20 seconds. So 20 plus 20 is 40. This should be 40. But obviously the distance is in micrometers. So yeah, it's just going to be 40. Because we're looking for the distance between the two chromatids, there are two of them. Okay, that's why we are adding two things up here. Okay, let's look at 1.1.9. Which one of the following indicates a type of variation for each of the human characteristics given? So in 1.1.9, the type of variation for each of the human characteristics given. Right, so now in 1.1.9, you'd be able to see that the height must be continuous, the skin color must be continuous, the earlobe types must be discontinuous, because now we're looking at which one of the following indicates the type of variation for each of the human characteristics given. Okay, so that becomes the option. The height is continuous, the skin color is continuous, the earlobe types are discontinuous, so it is option B. Now, okay, let's get the biological terms. You can get a term here that you can use. Okay, give the correct biological term for each of the following descriptions and whatnot. So yeah, let's discuss these biological terms so that you can remember some of them, because I can tell you that some of them are going to repeat. So if they repeat, then you're gonna be in a position to sort of understand them, and to be in a position to know what's happening there. So yeah, okay, yeah, in 1 second we shall just be having a discussion about these kinds of things there. um okay, right. Right, okay, we're discussing now. um all right, we're discussing in the next minute. I'm not going to keep you for long, because it's um it's just something that, okay, give the correct biological term for each of the following descriptions. Write only the term next to the thing, next to the question number, numbers 1.2.1 to 1.2.7. Let's just discuss this. The type of inheritance which produces an intermediate phenotype. Which one is this? The type of inheritance which produces an intermediate type phenotype. Which one is it? Right. So, right. What is it? Which one is the correct biological term? Okay. The type of inheritance which produces an intermediate phenotype is called the incomplete dominance. Incomplete dominance. Now, the point where adjacent chromatids overlap during crossing over, we just spoke about the chromatids now and the chromosome structure. But now, which one is it? The point where the adjacent chromatids overlap during crossing over is called the chiasma, or what we call the chiasmata. It's called the chiasma or the chiasmata. So this is the point where the adjacent chromatids overlap during crossing over. It's called the chiasma. If you want to call it the chiasma, you call it the chiasmata. The process whereby information is copied from DNA to RNA in the nucleus of a cell. The process whereby information is copied. So information is copied from RNA, from DNA to RNA. What is that process called? It's called transcription. So what is transcription? Is the process whereby information is copied from DNA to RNA. Is transcription. Okay, there's 1.2.4, the sugar found in a DNA molecule. What is that kind of sugar? What is the kind of sugar that is found in a DNA molecule? What do you call the sugar? Okay, the sugar is called the deoxyribose. Deoxyribose. The sugar found in a DNA molecule. What is the sugar found in a DNA molecule? It's called the deoxyribose. That is the sugar found in a DNA molecule. The bond that occurs between nitrogenous bases in a DNA molecule, there's a bond. The bond that occurs between nitrogenous bases in a DNA molecule. What is that bond called? Right, so let's look at the bond. The bond that occurs between nitrogenous bases in a DNA molecule is the hydrogen bond. It is the hydrogen bond. Right, is the bond that occurs between nitrogenous bases in a DNA molecule. The manipulation of an organism's genes to obtain a desired characteristic. So if you manipulate the genes of an organism to obtain a desired characteristic, what do you call the process whereby you just manipulate the organism's genes? What do you call that one? Right. What do you call the manipulation of organism's genes to obtain a desired characteristic? What is it called? It's called genetic engineering. Genetic engineering. It's called genetic engineering. The representation showing the arrangement of a diploid set of chromosomes. The representation showing the arrangement of a diploid set of chromosomes. What is this one called? The representation showing the arrangement of a diploid set of chromosomes. Representation showing the arrangement of a diploid set of chromosomes. What do you call that representation? It's called the karyotype. It's called the karyotype. It's called the karyotype. The representation showing the arrangement of a diploid set of chromosomes. Okay, I'm going to conclude. I'm not going to be very long. I'm just looking at the exam. Why am I looking at the exam? Because I just want you to note a thing or two. Some of them might just repeat, you know. Indicate whether each of the descriptions in column 1 apply to A only, B only. Okay, now this is very important because they like this. You write A only, or you indicate whether each of the descriptions in column 1 apply to A only, or you say B only, or you say both A and B, or none. But you don't just say A or B, just that. You must say A only or B only. So yeah, these kind of questions are going to be there at 9 today. None, okay, or none. Next to the question numbers 1.3.1 to 1.3.3 in the answer book. Source of variation in a species. Source of variation in a species, which one matches? Okay, so is it the random mating or random fertilization? The source of variation in a species. Obviously, you'd agree that here, this one is both. Is both A and B. Is both A and B, because we're looking at the source of variation in a species. It's both of random mating and also random fertilization. Both random fertilization becomes the source of variation in a species. Okay, now next. An organism possesses two factors which separate so that each gamete contains only one of these factors. An organism possesses two factors which separate so that each gamete contains only one of these factors. Now, the law of dominance or the principle of independent assortment. So which one is the correct one in 1.3.2? Well, the law of dominance or the principle of independent assortment, you realize that an organism possesses two factors which separate so that each gamete contains only one of these factors, or it's none of these. None of these. It's none of the law of dominance or the principle of independent assortment. Okay, if you look at the
reproductive isolating mechanism in species. Okay, now A refers to the breeding in the same season of the year. In fertile offspring, 1.3.3. Right, so which one is the reproductive isolating mechanism in species? In species, reproductive isolating mechanism in species. Right, so 1.3.3, you'll see that it is in fertile offspring. So it's B only. It's B only. So the reproductive isolating mechanism in species, it's not breeding in the same season of the year, but it's in fertile offspring. Infertile offspring refers to reproductive isolating mechanism in species. The diagram below represents a cell in early stage of meiosis. Right, so you have a cell in the early stage of meiosis. Right, give the following. Give the phase of meiosis represented. Okay, the phase of meiosis represented is what? In 1.4.1. So you'd understand that because of the nature of the representation of this cell in the early stage of meiosis, this is the early stage of meiosis, and therefore it's called the prophase 1. Prophase 1. Right, prophase 1. Number of chromatids shown. The number of the chromatids shown. Right, so you can see that the number of chromatids shown here, if you count very carefully, you realize that there are 12 of them. The 12, okay? Because now if you look at here, 2, 4, 6 by 2. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So the 12 of them. Okay. Now the number of homologous chromosome pairs, the number of homologous chromosome pairs. So obviously the chromosome pairs 1, 2, 3. So you look at the pair, it's just 2, 2, 2. So they're like 3 pairs. This is a pair, that's a pair, that's another pair. So there are 3 of them. The homologous series, number of homologous chromosome pairs, 3. Okay. Next. Identify the structure R. What is R called? What is R called? This dashed border, what is it called? Okay, this one is called the nuclear membrane. Nuclear membrane. So in other words, R is called the nuclear membrane. Next, what about R? What is S called? Because they're also asking S. 1.4.2. S, now this one, the solid line. Okay, the solid line is called the cell membrane. Cell membrane. If you don't want to call it the cell membrane, you call it the plasma lemma. Plasma lemma. If you don't want to call it the plasma lemma, you call it the plasma membrane. Call it the plasma membrane. Okay, plasma membrane or plasma lemma or cell membrane is what the outside is called. And then obviously the dashed one is called the nuclear membrane. Let's look at T now. T, what do you call this region space here? The space between the your chromosomes, what is it called? Okay, so this space T, 1.5 T is called the nucleoplasm. T is called the nucleoplasm. Okay. Nucleoplasm. Name 2 organs in an animal where meiosis occurs. Name 2 organisms in an animal where meiosis occurs in 1.4.3. Name 2 organisms in an animal where meiosis occur, or rather, name 2 organs. Name 2 organs in an animal where meiosis occurs. Okay, so right, meiosis occurs in the testes, but it also occurs in the ovaries as well. It occurs in the testes or it occurs in the ovaries as well. Okay, I'm not going to be long. I need to release you at some point. Right, so yeah, I'm just focusing on section A. The brinjal plant carries edible fruit. Scientists have been studying the inheritance of 2 genes, one for stem texture and the other for fruit shape. The stems can be smooth and or prickly, and while the fruit shape can be round, R, or elongated, small r. Okay, now name the type of cross that studies 2 characteristics. This type of cross that studies 2 characteristics. So this one here is called the dihybrid cross. Okay, name the type of cross that studies 2 characteristics. It's called dihybrid. Why is it called dihybrid? Because you're speaking of 2 characteristics. Okay. Now in 1.5.2, state the dominant characteristic for stem texture. Dominant characteristic for stem texture. The dominant characteristic for stem texture is, in this case, because the stems can be smooth, prickly, or that. Right, so obviously these scientists have been studying in terms of one of the texture of the other fruit shape. The stems can be smooth, prickly, or that. Okay, so the dominant characteristic here would be the smooth stem. Smooth stem. Right, recessive characteristic of for fruit shape. Recessive characteristic for fruit shape in 1.5.2 is the elongated fruit. Because the fruit shape can be round or elongated. Okay, so in other words, the recessive one is the elongated fruit. Elongated fruit. 1.5.3, give the genotype of a plant with a prickly stem and elongated fruit. What is the genotype? Right. What is the genotype? So the genotype would be NNRR, because the stems can be smooth or prickly, while the fruit shape can be round or elongated. So which means the genotype of a plant with a prickly stem, so prickly stem and elongated fruit. Okay, so it's going to be NNRR. Okay, because yeah, we're speaking of prickly, so it's NNRR elongated. So if you look at the phenotype of a plant with the genotype capital N, small n, double R capital, 1.5.3. Okay, so this one here is going to be what, 1.5.3. The phenotype of a plant with genotype NNRR. It's called the smooth stem, round fruit. A really smooth stem, round fruit. Okay, but because we're looking at the fact that you need to give the phenotype of a plant with a genotype NNRR, Okay, so it is a smooth stem, round fruit. Smooth stem, round fruit. So it is a phenotype of a plant with the genotype NNRR. The table below shows information on selected hominid fossils, and you have the common name of the fossil P, the tongue child, the species, the Australopithecus sediba. Okay, the tongue child species are the fossil site, the Malapa cave in the cradle of humankind, the Sterkfontein caves. Scientists responsible for discovery are Q and S, so we'll just fill in the table here. Okay, so let's look at this one. Okay, this one is very popular. I'm sure you know that very well. Name fossil P. Fossil P. Common name of the fossil P. The species, the Australopithecus sediba. Okay, those are the species. The fossil site is Malapa cave. What was found in Malapa cave? The common name of the fossil. The common name of the fossil is Karabo. Right, identify the species at R. The species at R for the tongue child. What species were there? For the tongue child. Okay, because now the common name of the fossil is the tongue child, but what are the species? The species are the
Africanus.
Yes, Australopithecus africanus. Okay, now we're finishing. We're just discussing this so that you can have an idea. And remember something, give the name of the scientist at, okay, what scientist is here? What scientist is linked to Karabo? And what scientist is linked to the tongue child? Okay, so we understand that linked to Karabo, the scientist is Q, and the scientist Q is, you can say Berger, but it's Lee Berger, or you can say, now this one, tongue child, who responsible for discovery? There's a scientist that discovered the tongue child in the Sterkfontein caves. What is the name of that scientist who discovered the tongue child in the Sterkfontein caves? Is that Raymond? The tongue child was discovered by Raymond. Dutt. Raymond Dutt discovered the tongue child at the Sterkfontein caves. Karabo at Malapa cave in the cradle of humankind. Karabo was discovered by what? By who? By Lee Berger. Let's just label these and then we're done. For example, if you look at section B, question 2, the diagram below represents a process that occurs during protein synthesis. You have this process, the diagram, and that shows the process there. Okay, let's answer some questions because this kind of structures repeat themselves very often, and they're going to be there in the exam. Identify molecule X. What is X called in this structure? X. What is X called? Because now you're looking at 2.1.1. Identify the molecule X, and then we're going to identify Z as well. 2.1.1a. Let's look at first X. What is X called? X is called the amino acid. It's called an amino acid. What about this one? Z. What is Z called here? It's called the messenger RNA, mRNA. This is the messenger RNA, but obviously this one is an amino acid. Okay, let's look at, give the nitrogenous base sequence of the DNA base triplet complementary to the middle codon on molecule Z. So on molecule Z, we need to identify the DNA base triplet. So now the DNA base triplet on codon or to the middle codon on molecule Z, right? So in 2.1.2, you realize that that will actually be the TAC, because you need to give the nitrogenous base sequence, right? So for Z, it's going to be actually the TAC. What about for Y? What is the nitrogenous base sequence for Y? Right, nitrogenous base sequence for Y is most certainly the GUA. Okay, you know these things because they're going to be there in the exam. So this is just a quick notes. Right, next. Right, name and describe the process shown in the diagram during the formation of a protein. Name and describe. So you need to name and describe the process shown in the diagram during the formation of a protein. Right, during the formation of a protein, the process shown in the diagram. Describe it. Okay, name and describe it. So obviously we can see that the process shown in the diagram, this diagram shows what we call translation. Diagram shows translation. But now let's describe translation for 7 marks. It's a lot of marks. So yeah, I'll just organize this quickly. What happens during translation? What happens during translation? So this one you just need to learn and memorize. Each transfer RNA, each tRNA carries a specific amino acid. Each transfer RNA carries a specific amino acid. Number 2, when the anticodon on the transfer RNA on the tRNA or the GUA. Okay, this is what happens during translation. Thirdly, you will have matches the codon, on the messenger RNA, on the mRNA. Yeah. Next, this happens all the time during translation. Okay, you don't have to say the messenger RNA or the CAU. The transfer RNA brings the required amino acid. The amino acid to the ribosome. The tRNA brings the required amino acid to the ribosome. This always happens during translation. Okay. Then the tRNA, the transfer RNA, brings the required amino acid to the ribosome. Amino acids. Amino acids become attached to each other. By what we call the peptide. We can say the peptide. peptide bonds. Amino acids become attached to each other by peptide or peptide bonds. Okay, but also during translation, one last thing. During translation, when this happens to form the required protein. So it's sort of the process involved, the required protein. All right. During translation, each transfer RNA carries a specific amino acid. When the anticodon on the transfer RNA or GUA matches the codon on the messenger RNA, then the transfer RNA brings the required amino acid to the ribosome. Amino acids become attached to each other by peptide or peptide bonds to form the required protein. 2.2. The table below shows the codons that code for some amino acid. Okay, so you have the mRNA codon, the amino acid. So AUG corresponds to the methionine, CAU to the histidine, the CUA to the leucine, the GUA to the valine, and so on. Okay. A mutation caused a DNA-based triplet to change from CTG to CTA. Describe the effect of this mutation on the protein formed. Codon. So this description is going to actually be as follows. So you'd say codon GAC on the mRNA, messenger RNA. Obviously, because this happened, this changed to GAU. Thirdly, both these codons goodness me, both these codons code for the same amino acid. Okay, they code for the same amino acid, or they code for the same aspartic acid. Aspartic acid. Okay, but also one last thing. Therefore, right here. Therefore, there will be no effect. Therefore, there will be no effect. No effect on the protein formed. All right. Okay, quickly. So, okay, the table below shows the codons that code for some amino acid. A mutation caused a DNA-based triplet to change from CTG to CTA. Describe the effect of this mutation on the protein formed. The effect of this mutation on the protein formed, the codon GAC on the mRNA, they actually changed to GAU. Both these codons code for the same amino acid, or the aspartic acid. Therefore, there will be no effect on the protein formed. Tabulate two differences between DNA and RNA nucleotides. Okay, what is the difference between DNA and RNA nucleotides? What is the difference? So I'm just going to do it here. DNA, RNA. What is the difference? DNA and RNA nucleotides. What is the difference? DNA has what that the other one does not have, because we're looking for differences. So we can think. Please come again.
Double helix.
Yes, you're on point. You're on point. This DNA has the deoxyribose sugar. This one does not have deoxyribose sugar, but it has ribose sugar. RNA has ribose sugar. What else does DNA have that the other one does not have? DNA has thymine or thymine, but it has what we call the nitrogenous base. Here's the nitrogenous base T. Okay, there's thymine or thymine, right? And obviously the RNA has uracil. If the DNA has thymine, this one has uracil. Okay, if you don't want to say it has uracil, it has the nitrogenous base U. When DNA has a nitrogenous base T, this one has nitrogenous base U. Next, 2.41. Okay, we're just running through these things here. Name in 2.41. That's what we want. Okay, we must first read. The Down syndrome is the result of an individual having, okay, the Down syndrome is going to be the end exam, but the Down syndrome is a result of an individual having an extra copy of chromosome 21. So in other words, if you have an extra copy of chromosome 21, then you have what you call the Down syndrome. Two genetic variations that can cause the Down syndrome are, what are the genetic variations? The trisomy 21 and the mosaic Down syndrome. All the somatic cells in an individual have 3 copies of chromosome 21 due to an abnormal process that occurs during gamete production. So this is just explaining to us why Down syndrome occurs. It occurs in those people, those individuals that have the extra copy of chromosome 1. Now, the 2 genetic variations that cause Down syndrome are, what are the genetic variations? Trisomy 21 and the mosaic Down syndrome. All the somatic cells in an individual have 3 copies of chromosome 21 due to abnormal process that occurs during the gamete production. Mosaic Down syndrome, the individual has only some cells with an extra copy of chromosome 21, which is caused by an abnormal process during cell division after fertilization. These are the 2 genetic variations. What are they? Trisomy 21, mosaic Down syndrome. Now, name the type of mutation that leads to trisomy 21. What mutation leads to trisomy 21 in those people that have Down syndrome? What is the mutation that leads to trisomy 21? Is the chromosomal mutation? Chromosomal mutation? Chromosomal mutation? Abnormal process during gamete production that leads to 3 copies of chromosome 21. What is the abnormal process during gamete production that leads to 3 copies of chromosome 21? What is that abnormal process? The abnormal process is non-disjunction. Right? The abnormal process is non-disjunction. The type of cell division that occurs after fertilization. What is the type of cell division that occurs after fertilization? It's called mitosis. So what is mitosis? Is the type of cell division that occurs after fertilization. That is mitosis. Next. Obviously, if you describe how the process in question 2.41b leads to trisomy 21, how does that lead to trisomy 21? 5 marks, it's a lot. Okay, I'm just going to give a sentence here. The chromosome write the chromosome pair stroke chromatids failed to separate. Failed to separate. Right? They failed to separate at position 21. During anaphase 1 or 2, they failed to separate during anaphase 1 or 2. So this is the what? It's the Down syndrome characterization. And so on and so forth. Okay, I can just continue with this. Describe two differences between trisomy 21 and mosaic Down syndrome. What is the difference between trisomy 21 and mosaic Down syndrome? What is the difference? Okay, it's just 4 marks. So let's just squeeze that one here. Trisomy 21 and the mosaic Down syndrome. What is the difference? Right, in trisomy 21, there is an extra chromosome. Or the 3 copies of chromosome 21 in each somatic cell. In mosaic Down syndrome, there's an extra chromosome only in some cells. The trisomy 21 occurs during meiosis. Trisomy 21 occurs during meiosis. Okay. The diagram below shows the interface of the inheritance of the Tay-Sachs, a rare disease, Tay-Sachs, a rare disease which leads to the destruction of neurons. It is inherited as an autosomal disorder controlled by 2 alleles, T, capital T and lowercase t. Okay, here's the question. Describe what is meant by autosomal disorder. What is autosomal disorder? We're just discussing what is it. 2 marks. What is autosomal disorder? Number 1, you must mention that if they ask autosomal disorder, is the disorder either disorder or the disorder is controlled by alleles, or you don't say by alleles, is controlled by genes, that. So it's one mark for the fact that the disorder is controlled by alleles or the genes that are located on the autosomes. The disorder is controlled by alleles or genes that are located on the autosomes. How many sons do individuals 1 and 2 have? How many sons do individuals 1 and 2 have? Right. So you can see the male without Tay-Sachs, and then you can see that the male with Tay-Sachs. So how many sons do individuals 1 and 2 have? Clearly, they actually have only a single son. So look at the diagram carefully. You can see, therefore, that they have only one son. Okay. I think I might just stop. I'll just stop because this is just about investigations and other things. Right, so I'll just continue, but now it's just about like 7 a.m. I know you need to bath and prepare yourself. I'll continue and discuss the rest of this. Yeah, but the paper is very short, generally. Okay, so yeah, but you need to bath now, I know, because it's like 7 a.m. You need to bath, you need to be at school by 8, 8:30. So yeah. I can just answer a couple of other questions, but I think I must thank you for joining us. I'm going to send a video so that you can just watch the things we discussed. I just wanted us to just say something about the kinds of things that are going to be coming, but this paper is going to be the sort of paper that is going to repeat very much so. It's going to be the prototype for this exam. So these kinds of things we discussed are things you really can expect to meet in the exam by and large. But thanks a lot for joining us. I would like to continue, but I think that time-wise is not a good idea to continue. You need to bath and prepare yourself. But thanks a lot, Tanula. Take your time, breathe, and do other things. Maybe memorize the last things you want to memorize, etc. Yeah, but thanks a lot and all the best. I'll send the video just now. Okay.
Okay.
Goodbye, Tanula.
Goodbye, sir.
Bye.